So I'm going to very briefly talk about the lagoon. I think everybody knows about it. I'll try to keep that short. The IRLO concept, and talk a little bit about some of the time series we've generated to date, some of our new LOBO work, the Indian River Lagoon Symposium, and some thoughts about the future. Again, since so many of you know the lagoon, and you're going to hear a number of talks, I'm not going to go into great detail about that. Suffice it to say, the lagoon is important to us locally, at the state level, and nationally, whether you look at the ecology or the environmental side of it, or the economic aspect. Also, most of you, of course, know all of the issues in the lagoon because they've been so highlighted in the past year by the media and others. These include water quantity and quality, pollutants of various types, impact on organisms, including things that are commercially important. The main goal of the Indian River Lagoon is to acquire and disseminate data and knowledge of the lagoon that's critical to its ecological function and sustainable management. In short, how does it work? How do we manage it? How can we optimize its use by everyone? There are three key components to IRLO. One is long-term, multidisciplinary ecosystem-based research. The second one is to use technology to develop advanced observing stations. And very importantly, the third one is collaboration among organizations. In terms of some of our long-term time series, our overall model that I've been working on for a number of years is basically what we do on land, or the loadings that we make into the water, impacts water quality. So our actions on land are directly linked to what goes on in the lagoon. And particularly, as you know, I work with marine plants, and seagrasses have been considered the most important organism for long-term management in the lagoon because of its critical role in terms of habitat and nursery functions for so many different animals. So just like I am today, it's really all about the fish, but the plants are really key to it. I started the time series actually almost 10 years ago with some state funding. We targeted the local area. The reason we did that, there was kind of a gap in knowledge, and in particularly what was the impacts of the discharges from the three main canals in Indian River County, North Main and South Relief Canal, that essentially drain most of the county. They were built 100 years ago so that we could live here, so that we could work here, so that we could farm here. And as we know now, there's been a lot of detrimental impacts on the estuaries. This is not unique to Indian River County. It's true throughout the lagoon. It's true throughout Florida. So our time series is using these instruments that have multiple sensors, in particular temperature, salinity, turbidity, pH, dissolved oxygen, chlorophyll. We have to pull these every week, download them, clean them. There's a lot of fouling issues in the lagoon. We replace them. At the same time, we do additional water quality measurements, especially on nutrients and suspended solids and color. We also measure light attenuation coefficients, which is basically the clarity of the water, how much light is actually getting to seagrasses. I could show you lots of data. I think the one that parameter that most people relate to the most is salinity. I think that's because of all the concerns about freshwater discharges. So this is the long-term record. This is the stations all plotted together. Looks kind of like a complicated matter, and it is. And you can see, though, that there's major events that happen. They might be hurricanes, and some of us have gone through them. The big hurricane started 10 years ago, started before this series, unfortunately. I don't have that data. But some of these have been historic events. Two of those events, for example, in 2007, 2008, those were all-time records for monthly precipitation. So again, what happens on land is intimately involved with what happens in the water. And we know long-term that about every 2.5 years, we're going to have a major tropical storm or hurricane that will come close enough to trigger one of these episodes. I think it's really important that we capture these types of episodes in long-term monitoring. We can make the data a little simpler for you if you'd like. This is the stations from north to south, is left to right through time, the different years. And you can see that when we started the work in 2005, it was actually the end of kind of a wet period. The hurricanes that happened the year before. It was a period of about 5 years of pretty heavy precipitation. Most of the time since then, we've actually been in an extended drought. So probably 7 of the last 10 years, our rainfall has been below average. We could simplify this even a little bit more and look at the spatial pattern from north to south, from Vero Beach south. And you can see there is a salinity gradient, as we would expect, as one goes away from the freshwater and discharges and gets closer to inlets at Fort Pierce. So we have this for a number of parameters. These are a subset that I usually like to focus on. But some of the major patterns are there is a nutrient gradient higher when it's related to freshwater discharges. Chlorophyll A, or pigment, what's in the water for the algae, also is higher at the southern end of where all that nutrients come from. And light attenuation coefficient, which is really important, I'll get back to. But first, let's talk about seagrasses. And I've already said why seagrasses are important. It's really all about the fish. Some people can tell you what the value of the lagoon is based on the acres of seagrass out there, and it's somewhere between $10,000 and $20,000 a year. Most of you know that in 2011, there was a catastrophic loss of seagrasses that was totally unprecedented. Unlike what we've seen in the St. Lucie estuary, that happens about every 5 years, what we saw last year. On average, it's been 5 years ever since I came here. But you can see here that we are a very interesting part of the lagoon where everything to the north of us had losses of seagrasses. In some cases, this is the 100% line right here. Whereas interestingly, to the south, we didn't have that impact. This is all due to an unprecedented event, the superbloom, so-called superbloom of 2011, by an alga that had never been seen or reported before in the lagoon. It's unprecedented because of the duration of the bloom. It was 8 months, and the levels of chlorophyll and number of cells. We've never seen anything like that. That resulted in about a 60% loss of seagrasses lagoon-wide in 1 or 2 years. Following years, you know, we had a brown tide, which is kind of a summer event, but most of the loss was due to that very first year. This is our local monitoring effort, our time series locally. And you can see as you go from north to south, this is Vero Beach. This is length of seagrass beds, basically how big are the seagrass beds. And you can see as you move away from Vero Beach, they are consistently quite larger. You can also see this is the period of the bloom. And we actually did have declines, sometimes very seriously, in the Vero Beach area. But interestingly, Harbor Branch, literally, this is Harbor Branch, Linkport. We're really right at the break-even point from where we saw what happened to the north and what happened to the south. So the monitoring work we're doing is at a really interesting part of the lagoon, which is being tapped into by a lot of people to try to explain what's happening both to the north and to the south of us. So seagrass presence and abundance is highly variable. It's related to water quality. There's only 7 species in the lagoon, only. But that's more than there's anywhere else in North America in any one place. So it's a very special place. These are the 3 most abundant ones. These are the 3 largest species. You can see again as you go from north to south along the top, the composition changes quite dramatically. And again, where we have more stable water quality, lower turbidity, lower water color, and so on, we have the higher abundance of species, and we have certain species that are found only when water quality is better. A lot of data here, but again, this is north to south always from top to bottom. This is through time over here. And I show this because you can see some very dynamic changes at some of these stations, especially during this period of greatest concern with the superbloom. You can also see some other areas that are more stable. This is twice a year, so you see there's an up and down also from summer to winter. In general, from north to south, seagrass beds not only get more expansive, but they get more abundant. This is the number, the cover within an area. So it's basically abundance within the sites. And you can see that decline in 2010 to 2012 better here than you can on the previous slide. And you can also see, I hope, that there has actually been recovery locally in 2013. We started our 2014 summer monitoring last Friday, and I'm pleased to report that at North Grand Harbor, the top one, for example, we've actually seen basically recovery to what we saw several years ago. So there has been some dramatic improvements that have happened in local waters. You can look at this in a lot of different statistical ways. This is a procedure called MDS, or multidimensional scaling. And you can use this to take long-term data sets. And basically what this is showing you is that the northern stations kind of all lumped together, but you can kind of tease them out a little bit there. But you can see they actually settle out very nicely along this water quality gradient. And statistically, we can look at changes through time. We can look at across stations, and we can look at across years. And both of them are highly significant. And we can talk about that a little bit more later. I want to focus on back to the fact that these are plants. What explains this overall pattern north to south from Vero Beach to Fort Pierce? Well, if you look at all the data, by far and away the thing that stands out is the light attenuation coefficient. So the bigger the number, the more light reduction. So it's kind of opposite what you think. So lower light is actually down here, higher light is down this way. So you can see as you get toward the inlet, we have a much greater availability of light. And that number is called the light attenuation coefficient. And here's that long-term time series for that. And you can see what happened in the superbloom period. Even though we were really not in the superbloom, we were on the periphery of it, the attenuation coefficient was markedly increased. And you can also see that as you go further south, there's much greater stability in the light field that the plants are receiving. If you look throughout the entire lagoon, a lot of the managers say anything above 1.5 is stressful. Okay. Anything, and you can see we hit that during that period of time, and that's when we had the greatest losses at those particular sites. And you can see we were at that level and seeing stresses here. I actually think when we continue the analysis of this, we're going to push that number down and make a recommendation that that number really needs to be like 1.3 or 1.2. That's really important because the managers need to have targets and they need to have actions that will get them there. I've also looked at what contributes to that light reduction. It's primarily suspended solids. There's particles in the water that you hear about, but chlorophyll A, or the pigment in the algae, obviously is a player, but usually secondarily. As you get further toward Fort Pierce, we actually see Taylor Creek influences. Those of you know locally, Taylor Creek. We're actually seeing color being the dominant attenuator. Most of you have heard about lobos. This is our next step. So last year we added into ERLO we added the lobo network. And this is basically our effort to expand our resolution primarily temporally, but also to some extent spatially. They have much better sensors that measure the same kinds of things I've been measuring for a number of years. But what's really cool about it is it happens in real time. It happens 24/7. We get a reading every hour. We don't have to go out every week. They can stay out a lot longer than that. And any of you can go to this website, or you can just Google Harbor Branch Lobo or Harbor Branch Water Quality or Harbor Branch Nitrogen, and you will find this, this site. And you can take a subset of the data. For example, this was last wet season, summertime. And you can see when you have reduced salinities, you have high color. So what? That high color is a sign of freshwater discharges because it's from decaying vegetation, freshwater and terrestrial. And it's a very, very important stress factor for the seagrass beds. And I don't think you need statistics to see what the obvious relationship is there. So what's the value of doing this 24/7? Well, this is 1 week of data. Normally we would go out and we sample a lot. Once a week sampling is a lot for nutrient samples. We did this as a comparison, and we just happened to go out that exact time where we had that peak for that particular week. But we could have gone out here, or we could have gone out there. What's the real number? Well, the real number is that there's a heck of a lot more variability than we're currently able to measure. And that's what this new technology is going to do. We have 168 data points now with a lot less work than we did to get one data point. So we're getting a lot more data. We're able to capture discrete events much better. That's going to help the models. You can go to the Harbor Branch website, Ocean Science Lecture, and get a whole hour tutorial if you'd like. Go to YouTube. Many of you were at the Inner Lagoon Symposium. I'm not going to talk about this very much. Suffice it to say, that's a very important event that we do every year. That is primarily how we are trying to extend collaboration throughout the lagoon to all science and all managers. We've had 3 of these. We're going to have another one next year. 300 people on average have come. This last year, we added a poster award, and I don't know if she's in the room, but her mentor certainly will be speaking in a few minutes. And we had actually 3 awards, and all of them were either Harbor Branch graduate students or hooked to Harbor Branch in some way. I want to just briefly mention leverage. What this project really has done, what the opportunity with SLP funding has really allowed us to do, is to demonstrate our ability to do long-term time series work. And that's what's really, really needed. Also now to be able to deploy this new technology, which is bringing unprecedented capabilities to the Indian River Lagoon. And the ability to bring people together in the Indian River Lagoon Symposium and in other forums. So we got kind of lucky maybe. Maybe it's all timing, who knows. So in the last year, I've gotten one project which is looking at the seagrass loss. We're looking at the recruitment side of things. We're tapping into our long-term data sets to see patterns, to see which species come back and how and why. Brian and Paul are both collaborators on this project that is just now starting, where we're looking specifically at the bloom areas to the north in terms of both nutrients in the macroalgae and in terms of what were the environmental stressors on those plants at that point in time. Most of you read about the foundation very generously asked us to submit a proposal so we could expand the lobe network. We're doing that. I think the equipment has been ordered. You know about $2 million that's coming to Harbor Branch to set up a lobe network specifically in the St. Lucie estuary. And we also got some funding from the St. Lucie Rivers issue team. So basically what I want to say is this is the way the lobe network will look in a year. So we will start from Harbor Branch. We'll be bracketing toward Vero Beach and Fort Pierce. We want to work also in Sebastian River inlet area. These are very dynamic areas of freshwater and saltwater. What we do on land when it comes in through the ocean, and also down the St. Lucie estuary. So here's some of the specifics. We have a meeting next week to work this out. Take-home message is the Indian Lagoon is very dynamic in time and space. There's a lot that has happened that we've done collectively through time. To really manage this, we need to understand all this dynamic aspects and pressures from the surrounding watershed. We need to understand that we don't really understand how things work. Ask anybody why we had that bloom in 2011. We don't really know yet. We have a lot of ideas. It's not worked out yet. So that's why we need the sustained focused research and monitoring, such as what we're doing in Erlo. And I would simply say that I think that we've really positioned ourselves now to be a leader and a catalyst for these efforts. I thank everybody, especially the folks who have worked with me for so many years, and the sponsors and the specialty license plate. I took the same amount of time over that you did.
Thank you, Paul, for that.